Welcome to today's USP and Pharmatech webinar, Cell and Gene Therapy Innovations to Drive Down Costs and Increase Efficiency. Thank you for taking time out of your busy schedules to join us. Next. Each registration is unique and grants the right for the individual registrant only to view this webinar. Sharing this webinar with unregistered viewers, either during or after its performance, violates USP's copyright and is prohibited. Next. If you have not already done so, we ask that you take some time to read the USP Education course disclaimer. You can download this presentation, including the disclaimer, from your USP Education account. Please follow the instructions in your confirmation email to download the presentation. Next. We have two speakers for today's webinar. Apoorv Puri, Principal Engineer, Product and Process Development, Pharmatech Associates, and Barrett Valentine, Director, Product and Process Development, Pharmatech Associates. Welcome and thank you to you both. I will now hand over the presentation to Mr. Puri.
Sure. Thank you very much. Hi all. Welcome today. We'll be kind of stepping through a few things, at least related to cell and gene therapy today, and then especially kind of what's been going on in the industry, and how people are kind of using kind of characterization approaches combined with a few other things to not only hopefully lower development time, but costs as well. But first, I kind of wanted to throw some, I guess, inspiration out there with this slide and, keep you guys grounded in the fact that a lot of the work done here today, and as is the case in science, there is no one breakthrough that really kind of, undoes any kind of current paradigm to make way for the future. It's all built on incremental understanding, and there's nothing you can really do. If there's anything I've learned about the last 10,000 years of human history, there's nothing really you can do alone. A lot of the work presented here hopefully lays a kind of solid foundation for the industry to move forward. So the agenda for today, I'll kind of discuss the scope here, and really kind of where my scope is a little bit limited with this presentation. So we'll be focusing primarily on cell and gene therapies, and then I'll dive into a little bit of the overview just to center you guys around what we're going to be talking about today, what the process looks like, maybe expected durations, and then really kind of lay the foundation for, why costs might be so high for certain therapies, and then really kind of discussing the costs and challenges. The high cost of this therapy typically leads to a lack of infrastructure that gets a little bit more pronounced when certain high costs are involved. And then a lot of these processes tend to be complicated, scale-out type processes. So really kind of knowing the challenges, knowing the entire process in and out, it goes a long way towards kind of the 5 major avenues for cell therapy cost reduction that we'll be talking about, which is contingent upon manufacturing success, lowering turnaround time, and so forth. And then I'll eventually present some closing thoughts, kind of where we hope the industry will head to not only make way for better standardization, but hopefully, more regulatory advancement of standards and best practices, and then, supporting new technologies and innovation. So the scope here today, largely we're concerned when we talk about cell and gene therapy, we're really concerned about the AAV lentivirus kind of portions, and then those lend themselves well to doing gene delivery in the adoptive cell therapy spaces as well. There are some more what I would call either rediscovered or uncovered technology. mRNA technology that's been used for the COVID vaccine's been around since the 80s, but finally now with the COVID vaccine, people are seeing the efficacy. And then we'll be kind of talking about some edge cases for technology as well. And then when I talk about adoptive cell therapy, really we're talking about CAR T cells, tumor infiltrating lymphocytes, and then more allogeneic therapies that are based on natural killer cells derived from either pluripotent stem cells or HSCTs. For the gene therapy process, really what you're always looking at is some sort of a working cell bank that kind of form or that are formed from the master cell bank. Typically, early phase development is kind of entirely focused on getting the cell bank to behave somewhat predictably. And here, kind of on the right, what you're seeing is an overall uh, flow of what this kind of gene therapy process really looks like, the manufacturing process. A lot of it is incremental expansion early on, which can take really anywhere from 7 to 12 days. To get the cells up to a working volume and a large enough viable cell density, and then ultimately there's some sort of transient transfection process that takes place that allows you to deliver the vector that you're looking for, and then eventually once your kind of cells reach a high enough population, uh, the culture gets incubated until your harvest criteria are kind of reached, and then from there on there's a bunch of downstream purification. Typically people will add a day or two for flexibility, and so any output coming from either your lysing process or your chromatography process might get stored, kind of further adding to the manufacturing time. And then once you have enough drug substance, essentially, then you're kind of working towards, final formulation, freeze down, and then really getting it, filled and labeled and packaged at that time. So that kind of gives you an idea for, where your current durations really come from, and then kind of how involved the process is. And then for the RNA conjugated LNPs, it's kind of a similar process. Really, your upstream processes are going to consist of some kind of culture, usually some sort of cell system that is, human embryonic kidney related, or whatever really works to develop your gene of interest or your RNA or DNA code that needs to be delivered. And then ultimately for RNA, a lot of the downstream process is focused on getting the right population of RNAs. So whether your RNA might be polyadenylated or capped at its termini, that really influences the way you end up capturing the RNA. And then similar to the gene therapy process you guys saw, this process can also last anywhere from 14 to 21 days. So already durations are really on the order of weeks, not necessarily days. And that typically, anytime you have the product in hand and not ready to go, either to be sent out for downstream CAR T applications, cell therapy applications, or some sort of direct gene delivery system, you're just essentially constantly adding costs. For adoptive cell therapy, things are a little bit different. So I'll be kind of discussing kind of technologies and approaches that seamlessly go back between gene therapy and adoptive cell therapy. So if something seems confusing right now, please do write down that question. We'll certainly try and answer it at the end. But for adoptive cell therapy, this really started with Steve Rosenberg in Bethesda, Maryland, in kind of the late 80s, early 90s, where they kind of discovered essentially that there's tumor infiltrating lymphocytes kind of local to tumors. So a lot of work was kind of done to, pursue solid tumor type of indications early on. Unfortunately, tumor infiltrating lymphocytes are still very hard to culture properly, and we'll talk about kind of the sources of variability. But the CAR T therapy that's kind of taken off since 2013 with several FDA approved therapies, that's gone through several iterations already. So your first generation CAR was really this extracellular single chain domain antibody, kind of hybridized with a transmembrane domain and an intracellular activating domain called CD3 gamma. But now you have these very complicated kind of fourth generation chimeric antigen receptor design with multiple co-stimulatory ligands. They can even induce cytokine secretion. So the technology has improved significantly just in the last 10 years. But even with cell therapy, the arm-to-arm kind of durations can be anywhere from weeks to almost a month. And that continuously, adds on cost, not only because currently it's kind of a very highly manual process, but ultimately, the big thing with CAR T therapy is that, a lot of the patients right now, none of the indications have CAR T therapy approved as a first line. So typically patients are kind of towards they have a very progressed disease state. So that not only is the time ticking, but we kind of also have this ethical quandary that we have to consider with patients that are on the clock. And then a little bit of a roadmap for you guys. I kind of already discussed, tumor infiltrating lymphocytes and kind of Steve Rosenberg starting that in the 90s essentially. But here are the things I kind of wanted to discuss really are, the prep regimen aspect, the fact that lymphodepletion and HSCT is still very much a part of the entire therapy makes the thing very staged. You do have to administer several rounds and different types of prep regimens and treatments to be able to have this be effective. Toxicity is kind of still a big problem. So this concept of, you're on target for the ligands you're targeting, but you're totally off the tumor indication. And cytokine release storms still continue to be kind of pretty major adverse reactions within the clinical space. And then not to mention, even between T cell receptors and CARs, there are still restrictions with the technology. So while chimeric antigen receptor technology compared to TILs has really advanced over the last 10 years, we're still not able to target non-solid tumors effectively. And then, not to mention those challenges, but we have the additional challenge of the cost of therapies. If you look at Kymriah and Yescarta, when Kymriah debuted, the cost was closer to, $475,000. Yescarta's still in the neighborhood of $200,000 plus. And then in the case of gene therapy, where Biogen's kind of developed Spinraza, the initial cost of dosing for Spinraza is close to $1 million. These are likely due to development, due to the complexity of the therapy, likely due to manufacturing failures that people experience while developing these technologies, and really, not only that, but the complexity of these processes really adds on. So since there aren't, when you talk about biologics, a lot of things in biologics these days are well automated, well understood, well characterized, but CAR T and cell therapies and gene therapies still are on the newer side. So the processes tend to be manual, at least in the cell therapy space, leading to much longer processing times. And then I've kind of talked about the ethical quandary before, but Corrales and colleagues, I believe at the MD Anderson Cancer Center, did kind of a survey of all CAR T dispensing clinics, basically. And right now, slots given per site per month, according to the survey that they did across 20 sites, is somewhere on the order of, potentially 0 to 4 patients. And on the average, generally one patient gets that slot per month. And then these patients could have spent anywhere from 3 to 8 months on the waiting list prior to apheresis. So we have this ethical challenge of, the cost is prohibitively high. The patient is essentially in a very progressed disease state and requiring some sort of therapy very quickly. So what can we do on the science aspect is kind of the questions we're asking today. And then lastly, I think one thing that we really want to talk about when we discuss cost reduction is platform technologies may be able to give you some relief from all these supply chain issues. Single-use items are still very high lead time. And then there's ultimately not enough CAR T capacity either on the supporting vendor side or the current manufacturers to address patient needs. So we do have quite a lot of challenges. So with that said, I'd like to introduce kind of the 5 major pillars for today. I guess you could call them, parts of the cog here. And really what we're looking to do is CAR T is kind of plagued with low manufacturing success. I keep harping on this concept of progressed disease states. Well, one consequence of that is that patients can typically have low acute lymphocyte counts, so low ALC counts. And ultimately, that doesn't allow you to AIF them in a way to isolate enough T cells to do the CAR T manufacturing process. And then ultimately, the cells show quite a bit of variability. With tumor infiltrating lymphocytes in particular, media tends to be a big concern. The tumor that was actually resected as part of TIL can definitely matter. So we'll kind of get into those avenues. And then some of the other things that I'm really concerned about is how do we shorten this duration that's on the order of weeks right now? So understanding cellular mechanics, understanding what is amenable to transduction, activation, and then in the case of cell and gene therapies, what is the kind of balance that you can reach as far as getting the higher viral genome titers while kind of controlling footprint and controlling the turnaround time. And then ultimately that'll lend us quite well to the low-cost viral vector production. Currently, the industry is dominated by lentivirus technologies, AAV technologies to deliver the target gene of interest, but there are other ways to do so. So we'll kind of talk about some CRISPR-Cas9 technologies, transposon electroporation, and then the last two that we'll kind of discuss are automation and modular platform technologies that can reduce error rates, really enable multiple parallel lot processing, and then hopefully, just kind of automate manual manufacturing to the point where you don't have to be so concerned about, re-cleaning the room prior to introducing another lot, or having chain of identity concerns. And then lastly, we'll briefly discuss NK cells as a possible other way to kind of uh, to potentially look at basically other ways to deliver the target gene of interest, or targeting the formation of T cells from basically stem cells in some sort of an allogeneic platform. So let's get into the manufacturing success portions first. I've kind of already mentioned some of these challenges in brief, but you'll kind of see uh, this is not meant to be comprehensive, I suppose. But on the left side, you'll really see kind of the challenges with tumor infiltrating lymphocytes. The primary mode of cell culture requires, this AB serum media, which is from basically healthy donors. But the AB serum can have very high variability, as you can likely guess. And then that can really present issues during tumor resectioning, regrowth, and then cell selection processes. It essentially leads to such a highly variable culture that it's hard to properly phenotypically isolate the right population of cells. And not to mention, since we're targeting solid tumors with the TIL therapies, surgeon experience is kind of a must. So the surgeon has to have a pretty good idea of how they're going to resection the tumor. You don't want too much fatty or necrotic tissue. The tumor microenvironment also dictates how many and how much of the TILs you're gonna isolate. And then ultimately, potentially, since your cell culture is so variable, addition of typically other antibodies or kind of CD137 types of surface markers can basically promote way better growth. And at the same time, this tumor microenvironment can be quite heterogeneous, which leads to a very large, diverse culture population. So these kind of challenges have remained with the TILs, and that's likely why you don't see it as an approved therapy currently. And then,
So I had a quick question for just
sure.
hopping in here. You mentioned on your previous slide, low LC counts can be an issue. I mean, at least this has to do more with CAR T, but what would be some more other, potential failure modes?
Yeah, so with CAR T, the other thing that really happens is you get these kind of terminally differentiated cells that are essentially they have exhaustion markers on them, likely due to the progression of the disease. It could be part of the apheresis process, but in my, kind of 4 years kind of wading through the challenges in this industry, I haven't really seen anything that suggests that. But the challenges are really kind of getting the right cell population. And so early on, phenotypic characterization can be kind of big because um, getting the right naive cells can actually be very beneficial because the naive cells are likely to persist longer when infused back into the patient. But then there's this challenge where terminally differentiated cells typically have higher cytotoxicity. So if they can target and find the right cancer cells, differentiated cells tend to be potentially more cytotoxic compared to the naive T cells. But at the same time, naive T cells that turn into memory cells, they tend to persist longer and promote a better response from patients. So, and I'll kind of get into that detail later, Barrett. So I'm kind of glad that you asked that question because that'll lead us pretty naturally kind of into the next sections. And then one of the last things is people will typically cryopreserve between unit operations. So for my adoptive cell therapy, if I have my incoming patient cells, I will typically cryopreserve them between steps. So let's say I get the AIF material. I will typically, resuspend that in media, and then for manufacturing flexibility, let's say I have, more folks at the first shift that can handle the next lot, then typically people will cryopreserve their cells. But even here, I think characterization is very important. One thing you do see is that cells do display a response to cryopreservation processes. Early characterization can go a long way towards making sure basically that there are no adverse conditions that arise as a result of cryopreserving cells. And then really for gene therapy, kind of stepping back a moment to the other half of this talk, is really, I would say, kind of five major areas of consideration. There's many more than this. I think having a trusted partner while you develop your cell bank and your early gene therapy AAV-LV process can kind of go a long way towards getting you a robust process that's, much easier to scale. And that kind of involves, having well-adapted host cell systems. That really, you have to start there. And if early development's kind of not focused on well-adapted cell systems, you can have these issues that become hard to explain, where the cells might be going through a lag phase when they should be going through a growth phase. And that might be due to a host of factors, one of which could be genomic stability. Compared to the 90s and now, genomic characterization, especially of cell systems that are bacterial in nature at least, can be pretty big in determining how stable that cell system is. So depending on the local environment, whether or not the bases A and T are highly represented in the genome, basically indicating higher genomic instability as compared to C and G, which are much more stable bases, that can give you pretty much an idea of how many passages can I ultimately run these cells through, and there's a ton of other ways that you can characterize that. But since genomic approaches have gotten so cheap, the industry for some reason still hasn't really caught up to that. While people still do kind of quantitative type PCR analyses, when I mention genomic analysis, I'm talking more about the overall composition, the overall tendency of the genome to continue to experience mutations, because especially in bacterial cell systems, it's not like mammalian cell systems, right? There's not necessarily these mutation correction mechanisms. And then one last thing I'll mention here that I think people frequently get confused on, especially when we're dealing with DNA, RNA types of, gene products, there is a balance between metabolism and gene replication that has to be reached. So there's frequently correlation between metabolic activity and gene replication, but it's not a linear correlation. So that means just because you're increasing your growth doesn't necessarily means you have a high vector copy number or a high plasmid number. So really looking early on, especially with potentially contract manufacturing partners, on how to optimize that culture, there's definitely techniques and other ways to kind of get there.
Just to follow up, I know Murphy's Law is always real when you're in a manufacturing setting. Have had any other experience with other additional challenges besides the ones you mentioned here with during manufacturing? Like,
You're talking specifically with AAV-LV?
yes, because I know those processes, at least on the regulatory side, are often very, very complex, and
Hmm.
so other things tend to come up. And I was just curious if you had experienced any other challenges.
Well, something we've seen in the past, at least for me, has been kind of this level of like version control and parameter characterization. So I'm zooming back even a bit further here, and we're really looking at the forest here. And so when I talk about process version control and characterization, I'm really talking about the early phase work that your development team might be doing, and really there, something that I've seen almost consistently is there is no characterization that is kind of thoroughly thought about. So when I say that, what I mean is uh, people aren't thinking in these factorial designs of experiments types of terms. So typically you have very many what I would call multivariate types of relationship between your reagent inputs and ultimately your process outputs. So something I've seen in the past that's been a gigantic challenge is just reagent variability leading to culture variability, leading to process variability, but nothing within your process design or development really ever, hinted at that. So, and that's either due to poor experimental design or really not understanding the ways in which kind of early phase development studies are kind of influenced by the reagents they use. So that's something I've seen all the time. It can lead to really strange clinical results because you essentially don't have a reason for why there's variability in your process. And then not to mention regulatory challenges there could be, threefold because not only now have you not characterized your system well enough, when that goes in front of a regulatory authority and they begin to question your clinical results and the way you've characterized your process, that could kill a very promising drug candidate right in the development phase. So next, we'll be kind of getting into the reducing turnaround time section of the talk. And really here, I wanted to give some other ideas and approaches. And really kind of the four that I came up with are the phenotypic characterization that I've been kind of harping on. So we'll get into a little bit of that. And then ultimately, maybe continuous process design is actually the way to reduce turnaround time. And this is not necessarily for adoptive cell therapies. I think we're actually a long way away from any sort of continuous process design there, not only because they're autologous, so that means one patient per lot, but even for the allogeneic therapies, there's just nothing that's really large scale and continuous that's been designed or developed yet. Some other things to actually think about are kind of rapid release testing. There have been a few methods that were developed in the late 90s, early 2000s to basically test for mycoplasma species, and then there are also other faster forms of sterility testing. Right now, sterility testing, if you're looking at myco, could take you up to 21 days. If you if you take the manufacturing process at 21 days, add another 21 to that, now you're basically at a month and a half, 6 to 7 weeks to even have a released drug product. And so once again, if we bring up those ethical challenges of, hey, we've got patients waiting for this, this becomes a pretty uncomfortable thing to think about. And lastly, what if we're talking about adoptive cell therapies, what if we could eliminate cell expansion? Because if you look at most kind of what I would say are now somewhat traditional manufacturing processes for adoptive cell therapies, ex vivo expansion is kind of a big deal, right? Because you're dealing with a patient with potentially low Lymphocyte counts, activation, transduction tend to be pretty harsh processes. So they can induce cell death in the cells you're dealing with, especially if they're exhausted or fully terminally differentiated. And then one last thing to kind of consider there is, well, I have to now expand my cell population, but that can typically add anywhere from 5 to 7 days. And then, unfortunately, if you do have something where you have an exhausted cell population, they might not even respond to ex vivo cell expansion that well.
You know, I did have a follow-up though. I think you can't have 2, 3, and 4 without number 1, right? So
I
you talk
suppose.
about the right cell population, you know, what ideally does that look like?
Sure. So you read my mind basically, and that'll kind of uh, transition us into the next slide quite well. So what I'd like to orient the presentees to today is some work that happened while I was at Bristol Myers Squibb, done by kind of the process development team between Lauren Brown and Jeff Teo. And really, we had some sort of an inkling that our manufacturing failures, essentially basically not reaching harvest criteria or not reaching your critical quality attributes, having something to do with incoming patient material. And our development team essentially ended up characterizing and publishing this work in February. And what you're looking at are the kind of CD8 positive cells and CD4 positive cells, and sort of this heat map of Spearman correlations. And Spearman correlations are essentially kind of non-parametric measures of ranks. So for the stats nerds out there, essentially what this is measuring is how, how well do two phenotypes, or I guess one phenotype or one quality of your incoming cells, how does this really correlate with kind of, some of the other uh, outgoing phenotypes of your modified CD8 positive, CAR positive T cells. So really what I'd like to focus the readers on here is your x-axis is kind of the incoming cell population, and then there's multiple phenotypes here listed with multiple surface markers. And what I kind of want to draw attention to, and the markers, the labels of the markers aren't so important, but know that CD45 positive markers and CCR positive, CCR7 positive is basically, kind of an indication of naive T cells. And if you remember, I've mentioned before that naive T cells will, when transduced with CAR positive, the CAR positive gene, um, they'll turn into effector memory cells, and they basically persist in the patient's bloodstream for a lot longer, and can have actually, uh, you know, uh, they can secrete more IL-2. There's all these kind of positive things that essentially happen when you control for your incoming cell population. A lot of manufacturing processes still kind of just focus on isolating basically all the CD8s and all the CD4s. But what their work kind of shows is if you're looking at naive T cells, well, they might be a lot better at, secreting IL-2s. Whereas if I'm looking at, differentiated, terminally exhausted T cells, they're a lot worse at, kind of secreting, IL-2. And that's just one thing to consider here. Uh, Differentiation states basically, at the end of the day, correlate with the final product profile, and they definitely influence how effective your drug is. And so that's something to that I'm hoping the industry kind of catches on. And this is not just their work. I believe, if I'm not mistaken, folks from Novartis in 2019 did something similar, where they've proven the same thing again and again, where incoming patient material, if you select for the right cells, can give you not only shortened manufacturing durations, but ultimately a better product profile as well, with higher TNF alpha secretion, higher IF and gamma, and typically any release assay right now in adoptive cell therapies looks at IF and gamma secretion. So the positive correlations here indicate that if there's a rise in, any of these x-axis population, it also gives a rise to certain other ones. But then you can also see these negative relationships where, terminally differentiated cells are kind of bad at, secreting the right cofactors and having the right activity. But unexpectedly, one marker of terminal differentiation is actually being CCR7 positive. Actually, I'm incorrect there. CCR7 negative and CD45RA positive. So let me see if I can find that on here. Uh, there we go. And this is really a marker for terminally differentiated cells. Sorry there. And if you correlate this with kind of IFN gamma, yeah, their cytolytic activity is high. But if you look at their IL-2 secretion or TNF alpha or other markers that would indicate a higher persistence time, you don't really see that. So there's all these other kind of factors that are negatively correlated. And then to switch gears again, once again, to disorient you folks a little bit, is kind of going back to the downstream purification processes for AAV, LV, and just gene therapies in general. One kind of technology that's been proposed over and over again in potentially in basically other industries that utilize chromatography for separations is this concept of kind of a quasi-continuous downstream process. Upstream, we kind of have perfusion cell cultures that steadily remove harvested cells at kind of a continuous rate using cell retention devices. But downstream is still this kind of in series blocky process that doesn't really enable you higher throughput. So one idea to potentially consider is to take that potential upstream clarified harvest lysate pool and can potentially design a process of what's called continuous countercurrent chromatography. And essentially what you're doing here is once you're done washing and equilibrating your first column for chromatography, and as you're essentially loading that washed and equilibrated column, you're washing and equilibrating a second column and getting it ready. And then these are essentially connected in series, and you can entirely automate this process. So between Delta V and potentially other automation platforms, if people were to develop kind of a well-characterized what I would call a custom system, it's certainly possible doing this. And essentially what this allows you is kind of greater resolution. So not only are you eluding your peak over much larger surface areas, but essentially you're also doing this process where you essentially have no downtime. So once your column 1 is eluded and your eluate is passing through to column 2, now you're washing and equilibrating column 1 again. And so you can essentially run this process as kind of a continuous series chromatography process. And this may give you a better alternative instead of kind of what's traditionally followed, where you might run anion exchange and then you store your eluate for the next day for processing. And then after that, you might run an affinity exchange column and then you store the eluate again for a further downstream polishing. If you were to just think maybe a little bit outside the box, it's certainly possible to generate somewhat of a quasi-continuous process.
So I just had a follow-up, and I'm generally used to a 3-column purification process. Granted, my background's primarily in antibodies, proteins.
Mm-hmm.
So are you saying that you could potentially eliminate polishing, or there should just be another hold and then you'd have to still have that polishing step?
That's a good question, Barrett. I haven't seen this specific iteration of continuous countercurrent chromatography kind of implemented at the industrial scale, but um, my thought process would be that if you just are a little bit more creative about the flow paths, so let's say you have anion exchange and affinity basically back to back in this kind of continuous countercurrent setup. In my mind, I don't know if it would be that much harder to basically, once your affinity and anion exchange are done at the levels that you want them. So basically you have the purity you need after those. While your downstream polishing steps is just a matter of adjusting your flow paths in such a way that once you're done with this portion of the chromatography steps, it would just be rerouting the downstream flow to your polishing steps. So whether that's ultrafiltration or you're doing something else entirely, I think it's still possible to integrate that into this design. So with that said, we'll kind of jump into the next thing, which is really the rapid release testing that I had been talking about. And really, if you look at the compendial methods on the left side, great, they're standardized, they're low cost generally, but there are some positive, some false negative, some false positive rates that do that do show across thousands and thousands of tests. And then ultimately, there might be some cross comparison required because if you look at the bacterial endotoxin method, there's technically three methods in there. There's gel blots, kinetic chromogenic methods. So let's say if one of your methods actually doesn't give you a conclusive result, you may have to go back and do some sort of cross comparison. And then, like I mentioned before, I mean, multiple culture conditions, multiple days of culturing, 21 days in the case of mycoplasma. It just this totally adds to cost because every day that this finished drug product sits in the manufacturer's hands is another day that somebody has to know where it is, what its testing status is, what its release status is, and that just continues to add on to cost. So something that the industry is doing, which you'll see this in adoptive cell therapies a lot, there were bacteremic culture methods called BacT-Alert that kind of debuted in 1997, but haven't really gained kind of a foothold until now. But the BacT-Alert is extremely low rates of false negative, false positive. Sampling is much easier. You can do sterile sampling, essentially a lot easier so you don't get these kind of false positive, false negative issues. And then not to mention the methods are just extremely quick. So the MycoSeq method is much quicker than 21 days. BacT-Alert is much quicker than the multiple days of incubation time. And then ultimately these can scale as well, because if you think about how many samples I have to culture in the compendial methods, that can also continuously add to your footprint. So if you're really trying to scale as an early phase organization, that becomes really difficult in those cases. And then the last one really that I kind of mentioned previously is potentially eliminating ex vivo expansion. So if we really look at the phenotypic characterization that's been done over and over again, and folks that are coming out with more and more results, there's a company called Graycell that basically has been trying to target 2-day or next day manufacturing of CAR T cell product. So that means very little cryopreservation, not almost, absolutely zero ex vivo cell expansion. And what they're really focused on is basically turning around the cells as quickly as possible so that they don't end up with, exhaustion markers or not, basically not amenable to cell culture conditions. And not to mention, this has enhanced effector function. So what I mean by that is, reduction of these kind of extensive ex vivo culture processes that may be leading to your T cells becoming exhausted. Well, these can also have other impacts, especially more T stem cell memory and just more efficacy of the drug product overall. And then since they are targeting kind of next day transduction and then this subsequent release, lack of this ex vivo culturing, and then really avenues of potential activation induced cell death are minimized. This can greatly improve manufacturing success rates, because manufacturing success rates, especially in the CAR T therapy, are anywhere from 60 to 75%. They are not 100%. So just because you have the patient's cells in hand, maybe you even have enough T cells, there's no guarantee that that you're going to have the success that you're aiming for. And then really a couple of things in quick succession that I'd like to discuss before we jump into the questions are, the current platform technologies that are available. If you were in the 90s right now, most of these would not exist. Unfortunately, with AAV-LV, you're never really going to get away from the large-scale primary culture cell systems, but there's all these continuous downstream technologies that are coming out, the periodic countercurrent chromatography that I kind of introduced, but Cytiva's got almost semi-fully automated ACTA columns, same thing with Sartorius, so these technologies could be big for the AAV-LV space. But really here, what's dominating is kind of the ubiquitousness of cell culture and cell enrichment platforms that are coming out. So that's pretty exciting. And then one thing I've mentioned that can really directly lead to more rapid release times in your kind of analytical process are all these other technologies that have come up. So ddPCR to do phenotyping is very fast. Rapid Myco, like I said, compared to 21 days is almost cut by 90%. And then you can also do inline cell measurement with a Visio, iLine F. So all these technologies can go a long way towards giving you better data sets, better characterization, shorter turnaround times. And the big thing is most of these are kind of single-use technologies on the right side. So not only is everything kind of hermetically self-contained, it's also in single-use platforms which are better for essentially cell culturing and other you know other considerations. So both gas exchange and potentially temperature control and other things in the case of incubators.
So there's a lot on this slide. So in your opinion, what do you think is one of the more promising platform technologies?
Sure. So the DynaSelect from Thermo Fisher has just recently come out. That allows you basically magnetically assisted cell sorting in a much more reduced platform. Same thing with the Thermo Fisher Rotea. I mean, your working volumes, in cell culture, at least around cell therapy, were on the order of like 500 mils to a liter. Even these are getting lower and lower and lower. So the footprint's getting so much lower with these devices that this is certainly a big deal for scale out, because if you talk about adoptive cell therapies, it's really actually very difficult to scale them out if you don't have the right considerations in mind, which include facility design as well, by the way, which I'll get into shortly. And then really the Miltenyi Prodigy is kind of changing the landscape a little bit, because not only does it offer magnetically assisted cell sorting, but it can incubate, it can do media exchange. It basically is the kind of the CPRO cell enrichment platform combined with the Miltenyi Clinimax, and it can basically kind of do your whole cell culture processing on one platform. So for certain clients, I mean, that might even be a better solution than anything listed here. So with that said, let's get into kind of the facility design portion things, and how to even maybe potentially automate the whole work process. So something that Solaris in California is currently doing is designing this cell shuttle type of a platform. It would be a fully automated process about the size of essentially a truck that can hold 10 lots. So you can kind of see that here. And then there's just a robotic arm that can basically do all the media exchanges, transduction, activation, whatever you need. And as you can see, I mean, this could be kind of a big deal in terms of, reducing manufacturing variability, more consistency, better traceability, and full automation, especially in these cases, can offer way better traceability than a, manual paper batch record with multiple operators.
I'm just going to assume that since this is so innovative and new, that Solaris absolutely collaborates with some of their clients to basically customize this to fit their process.
Yep, yeah, they're currently collaborating with Poseida Therapeutics, I believe, and some clients that are kind of early in clinical or potentially just phase one. They haven't cared to release the names of all their collaborators, but my guess would be this design is going to iterate over time, and definitely get better and better. And really, one last thing I'd like to quickly mention is, as you saw with the cell shuttle, if I can just do 10 lots in that cell shuttle that are fully self-contained, do I really need my traditional kind of a stick-built facility, right? So when I'm talking about stick-built facility, it's kind of every room has a purpose and an ISO designation, and you're really looking at things as like dedicated product spaces for this unit operation or dedicated product spaces for open vector processing. But as you survey the industry more and more, you realize that industries are kind of, especially big players like BMS, Novartis, people are opting for these kind of ballroom type modular panel system designs because it essentially offers them a way to rearrange the room without, committing so heavily to them. And then ultimately kind of give them a lot more flexibility than your traditional stick-built facility can. What you saw with the Solaris Cell Shuttle, I would say, is more of a fully modular system where basically you can kind of arrange that cell shuttle to do really anything within those 10 boxes. But the problem is the costs remain kind of prohibitively high still with those. So the cell shuttle might cost you, anywhere from $2 to $3 million. But I might just be able to build a facility for 1 million. So people might still be opting for stick-built modular designs, but as the cost of entry gets lower and lower, I would imagine that those technologies are going to outpace kind of your traditional facility, hopefully within the next 10 years or so.
So just looking at this slide, my guess would be that most companies are looking for something in the middle, like a modular panel system.
Yep.
Nothing fully modular, nothing stick built. But I mean, for the folks that do want to design these stick built things, besides cost, are there any other kind of typical delays besides the usual permitting issues?
Sure. So one thing we dealt with previously is, working for certain organizations where you might have open viral vector processing. The FDA is actually worried about cross-contamination in those cases. So let's say you do actually have a multi-product facility where you have multiple car products. The problem with the traditional facility design is now you have to have dedicated rooms for that open vector processing. So what I would suggest people do is really think about closing their processes with single-use technologies. So find weldable tubing systems with bags. And then the other thing is, which leads us right next to this next slide, is these kind of open ballroom designs. So all you see here are just kind of worktop spaces, but these can house everything from, benchtop incubators to your benchtop welders and sealers to kind of all the platform technologies I showed you guys earlier. So you could ultimately have multiple Clinimax systems set up and be able to easily process multiple lots within the same room. The issue becomes with your traditional facilities where you have dedicated product spaces is likely there's only enough space for one product in and one product out. And so your throughput considerations become a huge deal when you start talking about, well, what's the capacity of the facility? What's my basis of design? And what am I going to get out of it? And you'll typically find that the traditional kind of dedicated rooms with ISO classifications, those are maybe a thing of the past. I mean, especially with all these platform technologies that are such low scale, the emphasis is almost on jamming as much capacity as you can.
Have you heard of either, the EU or the FDA challenging aspects of facility design like this?
They will heavily challenge chain of identity concerns here. So let's say I do come in with a lot. I was supposed to work on this workstation here, call it number one, but somehow I got it mixed up with number two, and I now have the wrong patient cells or the wrong vector or just something else. So they will challenge you pretty heavily on how you mitigate the risks of, basically swapping the wrong product or maybe incubating the wrong incubator, or transducing the wrong lot with the wrong vector. So those things will be heavily challenged in those kind of open type designs. And then as we are running out of time, I'll speed things up a little bit. A couple of things that I did want to discuss were potentially low-cost vector sourcing because we really haven't talked about this. And ultimately, vector is probably one of the costliest things, both for AAV, LV types of things and ultimately CAR T applications as well. But if you guys remember earlier, I'd kind of talked about really nailing your process design to the point where you understand what your impact is based on reagents. You really understand what metabolic profile is going to give you the highest vector viral genome titers. And these really all need to be integrated during early phase development. And so when we're kind of choosing the manufacturing model, it's actually really important to think about, well, do I have the in-house expertise and the time to do this versus CDMOs? So having a trusted partner, especially early on, that realizes that there are going to be several challenges involved and actually has well adapted suspension cell systems, well adapted kind of culture methods, and they just really understand their cell lines. And if they do have to develop one, well, then they should have the expertise to help you develop the thing that you're looking to develop. And there's a ton of adaptive platform solutions out there. There's a company called PolyPlus that basically has ready-made transfection platforms. I highly encourage early phase companies to really look at those before really committing to a specific cell system or a specific CDMO, and really look at the breadth that's out there, because even 10 years ago, there just weren't this many platform technologies available. And then last couple of things I will kind of leave you with are these transposon type technologies and potentially CRISPR-Cas9. These technologies were basically not discovered on accident, but the point here is that, these were developed from systems, not from the mammalian genome. So these are kind of synthetic sequences that were found to be moving or translocating within the genome. And essentially what happens is there's this kind of variable segment of DNA, and ultimately there's something that drives the transposase to attach to these sites, which are called there's basically these repeats that the transposase recognizes, and ultimately ends up cleaving the gene and displacing it somewhere else within the genome. So I think these can be very powerful technologies for gene delivery, whether in CAR T, whether in gene therapies. I think these have a ton of promise. Now, one risk is that the integration can be kind of variable. So if you're activating oncogenes while doing, transposon type of gene delivery, well, that can be kind of definitely an unintended consequence. So as the kind of industry develops, my expectation for this technology would be that we have even better characterization methods or highly engineered transposons sequences that can basically dictate very targeted integration. And CRISPR-Cas9 is similar. While the mechanics are different, the essential principle is the same, where you're taking, the intended sequence that you want to integrate, kind of conjugating it with this Cas9 linker loop, with a tracer RNA and a SG guide RNA. And ultimately, this can have much better targeted, much better genome integration. But random integration has been observed with this technology as well. So if people are pursuing this, my expectation really here to drive down the risk and to reduce cost is to really characterize the integration as best as possible. Like I said, there's a ton of sequencing techniques that can help you, really characterize where and when that integration is happening. So as these, methods become more and more recognized, I think these could be valid analytical methods in the future to test, genome integration.
I mean, I agree that this might be the future, but you touched on it yourself. I mean, I think safety is key, and especially since you don't know the long-term effects. At least with CRISPR, you're hearing a lot out there that
Yep,
people are
yep.
doing. That seems a little Wild West to me.
Yeah, and I think I think without those proper characterization types of studies, I even think the regulatory authorities are going to kind of balk at the technology and say, well, you need to prove the safety and efficacy because the risks are likely too high. And one of the last modalities I'll kind of discuss now is the NK cells, really kind of uh, the whole premise here is that NK cells compared to CAR T cells can be much more. They basically display way more tumor potent, like way more potent anti-tumor activity. And essentially the way they do that is because they can recognize either the absence of MHC class 1 kind of receptors or have multiple activating receptors and ligands on the cell surface, which they kind of use to recognize either a stressed or a damaged cell or a tumor cell. So people have been using kind of induced pluripotent stem cells, giving them the right signals to form them into NK cells, and even conjugating certain NK cells with, TCR receptors or CAR-like receptors to basically make them, highly targeted, focused homing missiles. This is something along with the transposon sleeping beauty platform that Alaunos Therapeutics is pursuing, where they're basically kind of combining everything into this next day manufacturing process, and going towards a way to use a different modality essentially to target cancer cells. I have frozen here. And really the last things I'll leave you with is kind of potential advantage of this. Big thing with NK cells is effector function. You can induce a much better effector function through kind of germline encoded receptors. And the tumor targeting for CAR NK cells has been pretty promising for a while. And one another kind of consequence of that is just reduction in cytopenia. So for patients, this is a very much a welcome change in technology. And really closing thoughts I'll leave you with kind of the 5 areas we've discussed. We really discussed how to kind of increase manufacturing success through really looking at phenotypic characterization, some low-cost vector approaches. And then these are kind of, the one thing I will emphasize here, these are all interrelated. So your science organization, your development, your manufacturing organizations really need to work in tandem to pursue all of these. And I'm not saying one is necessarily better over the other, but there's so much out there that I think it's easy to pursue just one, thinking that it's going to work. But I think pursuing multiple ways to reduce costs can be pretty beneficial, not only in basically bettering your product profile, but ultimately providing a product that's that's going to be efficacious for your patient. And what we'd really like to see, both from, I guess, an industry perspective and a long-term perspective, is really for the science organizations in early stage, especially for clinical phase company, to kind of maintain that cohesive structure and really encourage kind of holistic thinking, both from process characterization and development, and then ultimately, your practice as well. And then one last thing I'll leave you guys with is, none of this really changes until the industry decides to expand standards and pursue these technologies. I think information sharing right now is better than ever. People are actually publishing what they find to be, major improvements in their process, which wasn't necessarily the case 25, 30 years ago. And really this, expanding of these standards can lead to more integrated thinking because people are thinking about all these early phase concepts. So there are valid concerns regarding the side effects of CAR T-cell therapy, such as CRS, neurotoxicity. In view of this, why is there not more focus on NK cells for tumor therapy? That's an excellent question. So the effector function for NK cells kind of dictates how long they stay, intravenously when administered. The other problem actually is that NK cells will undergo like apoptosis or whatever if they don't have other co-stimulatory signals. So with NK cells, it's like actually pretty important that they get some sort of steady signaling of cytokines. And so, in the case of like Alaunos and these other kind of companies that are pursuing this NK cell technology, what they're looking to do is basically integrate the secretion of that cytokine into the engineered NK cell. So that could be pretty promising in the future.